3 minutes after. Good afternoon. I'm Angela Padilla. I am the director of the Skirball Science Learning Center. I'm an adjunct in the Physics and Astronomy department, and I'm finishing up a second master's at City College, and I'm doing research in satellite imagery for inundation, something or other. You'll hear more about that in a second, and I will pop on to Ines, but I'm so happy to be here. So thank you, everyone, and we're just rolling with the punches.
Hi, I'm Ines Miyares. I'm a geographer in the Department of Geography and Environmental Science. And my work is in tourism geographies, the intersection of tourism geographies and settler colonialism. And I will popcorn to Amber.
Hi, everybody. I am Amber Martin, and I'm in the Department of Psychology. My research is broadly, just to touch on it broadly, my research is looking at deaf children's language acquisition and how both internal systems work, the acquiring of language for children, and also now I'm starting to consider and look at the broader systems of family, community, and global systems, and really look at how that increasing interconnectivity of the world, how we can look at that and track that global systems all the way down to the internal and how it impacts deaf children's language acquisition. And, oh, sorry, I will popcorn to who hasn't gone? I'm not sure. Is it just us who you want to introduce, or can I select anybody?
Randye's great, and then I'll have Randye popcorn to Carmen.
Sure. So popcorn to Randye.
Hi, I am Randye Rutberg. I'm in the Department of Geography and Environmental Studies. I'm on the environmental studies side of the department, and I'm a geochemist. I study carbon and climate, and that's what I'll be talking about today. Carmen?
Hi, I'm Carmen Melendez-Vasquez. I'm in the biology department. I'm a glial cell biologist, and I will tell you a little bit more about that in a minute.
Amazing. Thank you all so much. And Angela, if you want to start us off.
Okay, I will start us off. I can share, right? All right. And let's see my slideshow from the beginning. Oh, wait, first let me share first before I do that. Zoom. Share. And I guess my whole this here. Are we seeing? Nope, wrong one. Is this the right one? Maybe. Yep, this is it. Good afternoon. So as we said before, I'm Angela Padilla. I am working on Wheeler Marsh backscatter, and to talk about it, it's our area of interest. It's in Wheeler Marsh, Milford, Connecticut, just at the Long Island Sound. So this is Wheeler Marsh here, and it's a wetland area. And we'll talk more about that. And in context to where we are, we're somewhere here. There's Port Jefferson and Long Island, and then you go across the waterway, and there is Wheeler Marsh. It's smaller than Central Park, but it's still significant as far as marsh area goes. And okay, so it sort of looks like that. I always like pictures to give people context, like why are we doing things in the first place and what are you talking about? So my main thing is to classify wetland inundation in Wheeler Marsh at Milford, Connecticut. It's about 23-acre barrier marsh at the northern border of Long Island Sound, next to the Housatonic River, right? It's bordered by that. So we're using radar data, and we're using L-band, L for long wave, different wavelengths of light. Like most of us know the spectrum, all the colors. But there's UV, there's infrared and radio waves and all those types of things. So we're focusing on microwaves, which are just above radio waves. They're kind of long. And this helps penetrate vegetation. Why do we use radar data in the first place? Because it can see through clouds and rain, and you can still see things. And what I mean see, it's basically a response mechanism. So like when you shine light on something and how bright the light shines back at you gives you some information about that whatever surface you're shining light on. So same concept, but using things that our eyes can't detect. These are things that other special sensors can detect. Our eyes can only detect the visible range. So without further ado, so it's effective at capturing backscatter from what I call short grass, because Spartina alterniflora is difficult for me to say. Hence, I was never a biology major. Everything is so complicated with biology. So, and this is what it looks like. Just tall, I mean, not tall, short cordgrass. And we'll talk, so this is what it looks like. And I generated something with ChatGPT. I like using ChatGPT. Has been tremendous with helping me try to re-explain and fight when I don't understand things. You know, again, it's not the end all be all, but if you know something, ChatGPT helps you get your context and organization when nobody's up at 2 o'clock in the morning working with you. So again, radar data. L-band, talked about that. And this is the spectrum. This is the ENM spectrum, the part that we can see. So I'm just giving you more context. This is the full spectrum from radio to gamma rays, right? We know gamma rays and X-rays and UVs are harmful. This is our visible infrared microwave radio. And that's what we're talking about, light waves. Okay, so study aim. To validate classification methods in anticipation of NISAR's goals of global observations. NISAR was just launched in July. Now, this is a great thing for everybody who's been following the news, because now it's science. You know, science has been so under attack with our current administration. But the fact that India and America came together to put this satellite up, I think anything else doesn't really matter. So that's like really exciting to me in that way. But it's a new satellite. They have other methods, but what I'm trying to do is understand how the methods work in the first place so that I can later on validate NISAR data, which is starting to come in soon, but it still has to be processed. So a lot of this is me learning so that I can learn, FYI. So you want to validate classification methods in anticipation of NISAR, and then provide, because NISAR with the satellite data is going to be improved temporal coverage. It's going to be going around the whole globe, not just little pieces, because a lot of the data that we have for radar is with the air flight data. So they're only covering small parts. Here we want to get the whole image, but we need some context to make sure that what we're looking at is correct and is it improved. That's the hope with the satellite data, with regular airplane data. High resolution wetland mapping, but the status is there's still no results yet. I'm still learning. I'm still working things out, but I've made some nice, that's the satellite NISAR. Some improvements with my learning mechanisms. So yay to NASA. So the goal is to establish reliable methods and the broader impact. And this is where the LUCE scholarship comes in. We're a service, right? Why do we want to do this anyway? You want to support understanding of ecosystem services at small coastal sites, including, right, flood attenuation. That's why we have these barrier islands, because it helps us from getting flooded inland, right? And if you're taking care of this stuff, then we have a little more protection. Habitat provision, there's hundreds of birds that migrate at Wheeler Marsh, and you want to make sure they do this. And then, of course, always recreational value, not just at Wheeler Marsh, but other wetlands, across the country. So this is what I've gotten so far. And, you're just seeing a bunch of maps, and, pay attention to that pork chop area that I call. It looks like a pork chop, right? But this is part of the Wheeler Marsh area. This part here where the pork chop is Nels Island, and it's also around water. Now, this dark area, what you're seeing is backscatter that's not being backscattered. That's like shining a light and not getting a response, like when you see black, because it's absorbing all of that. That when it absorbs all the light and doesn't shine it back to you, that's indication that it's water. So when it's bright, It could be grass, it could be a building, something like that. So the bright spots are the parts that have less water or are not inundated by water. The dark parts are all inundated by water. That's the main thing. And what you see with all this dark spot, oh, this is mostly water except for that pork chop area, right? Well, this is the rainy season. This is what we would call high tide. There's a lot of water, and this is May 7th of 2023. This is the same area, just 3 weeks prior, where you see a lot of the brightness, just 3 weeks, and it's a huge difference. That this is above, that the cordgrass is above the water, and all these little dark parts, it sort of looks like a piece of steak. Those dark parts are part of the river and waterways. So that's what you're looking for. So this is April 23rd, 2023. And you can see what you're seeing here is, now this is more about polarization. When you're standing at the beach and you see the water wave coming at you, right? You see it go up and down. It's vertically polarized. So you're going to do this. Had it been horizontally polarized, you would put your arms out to the side so that you don't go this way. So polarization just means the direction of the way the light is going. So this was like the big thing for me. And what we're looking at is standard UAVSAR data. So this is standard radar data. They also have radar data that's simulated to look like NISAR so we can know what to expect. So I don't have NISAR data yet. So this was me playing around with regular radar data. And then when you see later on, so this is the acquisition dates and all the fancy stuff, then we're going to compare it with NISAR data, or NISAR simulated data, how shall I say. You see this? This is NISAR simulated data. How many minutes do I have?
You have about 1 or 2 minutes to wrap.
That's fine. That's all I need. This grainier area, this is the NISAR simulated data. It's a lot less data points. So it's not as sharp, it's not as clear, but that's okay that I got this far. So you see NISAR simulated data, and this is actual real radar data, but this is promising that I know what I'm doing, that I know how to map this and take raw binary data and try to make some sort of comparison. That's really so far where I'm at, and learning how to use all these tools, because I had to learn how to use Jupyter notebooks. These are hubs for me to learn Python and mapping tools like GIS, QGIS. I use the free band. I like to use open resource things. And then when we're looking at the brightness, what we call the backscatter, here you see it as a map. Here, what these histograms show is that just the total, what the values that I'm looking at. And again, what you see here are the totals for one date versus the other date, high tide versus low tide. Now you can see how really different the NISAR simulated data is versus the standard data. A lot more data points for the standard, a lot less simulated. So you can't directly compare them, so you have to normalize them. And then you get things like this. So the ones in color are the simulated data. The ones that are in grayscale, those are the standard data. So now I know what I'm working with and how much I can actually compare. So that's really what I'm trying to do right now, is learn how these things function so that I can say something later in the bigger picture. So I can talk forever. This is more, again, this is the the high tide, low tide, and then I'll keep going. And then again, now we have to look at digital elevation models, and then just looking at water versus no water. If it's no water, it's green. If it's water, it's blue, and again, I'm looking at it at different angles so that I can build a better picture. And hopefully that you guys understand that I'm doing this for you, for us, just to see what's going on. Because not only is this good for wetlands, but also agricultural, flash fires, what do you call those things? Forest fires, all types of things, volcanoes, eruptions, and things like that. And of course, I have to go to other sources and look at what the water levels are. So I'm looking at a website that tells me the water levels. So I'm really understanding that what I'm saying makes sense. So that's it for now. I can talk forever. You guys know I can. But I'm going to end here. So thank you so much. And I wanted to be respectful of everyone's time. So help me stop this share. Okay. I have to figure out how to stop it. And somebody stop me from sharing because I can't. Oh, wait, I found it. Thank you so much, guys.
Amazing. Thank you, Angela. That was so interesting.
Thank you.
I think you're muted.
Yes, okay. No, just figuring out once I shared how to unmute. Okay, so I'm looking at how museums and other heritage sites, botanical gardens that are linked to heritage sites tell the story of land acquisition, of the story, the backstories. And so I'm going to start with this one quote, that the social interpretation of the past is constituted in part through a construction of material sites of memory, such as museums. And then the geography of memory serves as both the medium for representing memory, and because of its own complex material history, the very subject of what is remembered or forgotten. So I've been looking at the National Tropical Botanical Gardens. There are 5 of them, 1 in Florida, but 4 of them in Hawaii, and I've been focusing on the ones in Hawaii. The 2 main ones, McBride and Allerton, are on the south shore of Kauai, and then there are 2 others, one Limahuli on the north shore of Kauai, and Kahanu on Maui. The garden at Allerton was a privately owned estate that really pours out the opulence. It was an artist who developed it. Almost all the plants you see here in this garden are introduced, and the tour gives very few hints as to how this land came into ownership by Allerton. But it mostly focuses on the beauty, the waterworks, the artistic, without really giving any sort of sense as to the settler colonial legacy of the displacement, the takeover of the land, first by Sugar Corporation and then being sold to Allerton. And this is the most expensive of the gardens to visit, but the fees go to support this research center that is the largest tropical seed bank in the world, that is focused on protecting endangered tropical species from all over the world and attempting to reintroduce endangered and essentially on the verge of extinction plants back into a tropical environment. It also, these higher fees help support the 2 lower fee sites, the Limahuli Garden, that is actually an archaeological site and has multiple elevations to it. So it's where they're reintroducing a lot of these plants that are being studied at the research center. So the fees, they help protect this archaeological site. And on Maui, the Pi'ilani Heiau, a heiau is a sacred space. It's the largest heiau in all of Polynesia, remaining heiau. So, these gardens also have the tourism story, but these actually tell a more accurate story. Because they're archaeological sites, they're funded heavily by the other one that doesn't tell the story, that most, you end up with what's been called a paradoxical, it's essentially a paradox to be needing to, you only want to tell so much without offending the tourist. So then now I'm doing work on museums and how the legacy of the sugar plantation era is being told, and going to these museums to see the voice that is given and how the story of the past. So here's an example of one of the museums that I've been studying, the Alexander and Baldwin Sugar Museum. Alexander and Baldwin, they were one of the big sugar corporations. So it's telling the story from their voice as development heroes. We came in and did all this wonderful stuff to raise up the Hawaiian people in place, and how they tell the story is very different from how museums that tell it from the voice of the laborers. So for example, some of the language I found, here they're describing plantation communities that opening up virgin lands for cane cultivation. There were no virgin lands, they just weren't in cane cultivation. All land in Hawaii was partitioned and under cultivation by someone in their social hierarchy before the settler colonial era. Another quote, one of the things that was practiced in order to keep the laborers from uniting and organizing for more pay, was to segregate them, and they would pay them differentially based on their ethnicity. And so, here they've got this little quote that kind of mentions it, but, kind of whitewashes the real legacy of ethnic segregation and differential treatment to discourage cooperation among ethnic groups in the event of labor dissatisfaction, and to ease transition to the new land by allowing immigrants to live among their fellow countrymen. In other words, we were being good to our immigrant laborers by segregating them. And so it's telling the story, but in a way that makes the corporation look like a hero. So I've been, visiting multiple museums to see how they tell this story. And here's another quote: that tourism landscapes are constructed and marketed in selected ways that reaffirm long-standing patterns of social power and inequality, and thus influence whose histories and identities are remembered and forgotten. So, thinking about how this applies beyond my own research, especially applications to the classroom. There are a lot of us who incorporate museum visits and other site visits into our teaching. And the typical museum visitor, when seeing a display, experiences a 3-second, what's called a museum gaze, where you just kind of glance at the whatever a material, cultural thing or scientific thing is being presented at that museum, and that we really need to challenge our students to move past that museum gaze, to critically think about what they're seeing and hearing, and to think about what aspect of the story is not being presented, to move past just trusting the curator to have told the complete story. And with that, I will stop my share and popcorn it over to Amber.
Randy's next. Yep. No worries. That was so, you all are taking us to some amazing places. I'm so excited. Thank you. Thank you. I think I am guilty of the museum gaze, I will tell you, and I'll rethink that.
Okay, good afternoon, everybody. Want to make sure you can hear me first. Yes, okay. First of all, thank you so much for convening this session. It's really a pleasure to hear what everybody's working on. The title of my talk is The Carbon Cycle and Climate Change on Decadal, Millennial, and Glacial Timescale. The overarching question of all of this work is, what are the controls on carbon cycling on different timescales, and what are the impacts on climate? So the key questions that I'm going to talk about today are, what is the impact of orbital forcing on glacial interglacial changes and ocean and atmospheric carbon dioxide? What forcings influence the carbon cycle on millennial timescales? And a more recent project are invasive plant species influencing the growth rate of canopy trees in temperate forests, and by extension, their ability to store carbon. So you'll see I'm working on hundreds of thousands of years timescales, 1,000-year timescales, and decadal timescales. CO2 is one of the most important greenhouse gases on the planet. In the absence of anthropogenic inputs, carbon cycles between different reservoirs in the Earth system. And that's why I'm showing, that's the point of this diagram here. This is a simplified version of the carbon cycle. Natural changes in atmospheric CO2 concentrations were relatively slow and have acted as feedbacks in the Earth system through time. And they have either amplified or damped natural climate changes. But today we are running a real-time experiment. We're adding carbon dioxide at an unprecedented rate and changing the Earth's climate. So studying the carbon cycle in the past can give us insight into the mechanisms that control fluxes into and out of the atmosphere, and this knowledge is important for projections of future climate change. Today, I'm going to focus on ocean uptake in two of my projects, and photosynthesis and the growth of trees in the third project. The Earth's climate has tipped into and out of glacial periods every 100,000 years for the last 600,000 years. Prior to that, the Earth cycled into and out of glacial periods every 41,000 years for the past 2 million years. This is an image of the glacial world. And the key idea here is that during these glacial periods, there was far more ice on the surface of the planet. And here we can focus on the Northern Hemisphere and see the large Laurentide ice sheet and the Greenland ice sheet and the Fenno-Scandinavian ice sheet that covered large portions of North America and Europe. These glacial periods are characterized by extensive continental ice and concomitant changes in ocean circulation and the hydrological cycle. I want to just take a moment and say that Hunter College would be under a mile thick slab of ice 20,000 years ago during the maximum of the last ice age. The agreed upon mechanism that drives these cycles are subtle changes in the Earth's orbital configuration due to the gravitational pull of our largest planetary neighbors. This causes fluctuations in the amount of solar radiation that reaches the high latitude Northern Hemisphere, and this initiates the growth and decay of continental ice sheets. However, the change in radiation itself is not enough to cause these ice ages. We also know from ice cores that atmospheric CO2 increase during warm periods and decrease during cold periods. This is a natural phenomenon, and we don't know the exact mechanism that causes it. We do know that on this timescale, changes in the flux between the ocean and atmosphere drive the changes in the atmospheric concentration. However, the exact mechanism has not been identified. This is an image that just shows how atmospheric CO2 has gone up and down. Each of these peaks represents a warm period in the Earth's history, and the valley represents a cold period. And the blue line represents CO2 variability. These are natural fluctuations that occur along with the ice ages. Below, we have a record of temperature in Antarctica as a proxy for global temperature. Where it's high, it's warm. Where it's low, it's cold. And you can see by looking at the pattern that the carbon dioxide and the Earth's climate change together. So my research focuses on the ocean's role in this process. How is the ocean changing to absorb CO2 during cold periods and release it during warm periods? So I use climate model simulations of different orbital conditions to try and ask the following questions. Can we identify dynamical changes in ocean circulation that are consistent with the glacial sequestration of CO2 in the ocean? What is the relative impact of changes in tilt versus changes in direction of the Earth's axis? And which orbital configuration dominates the entire Earth response during the coldest parts of the glaciations? And so far, my initial conclusion is that glacial precessional parameters contribute to the storage of carbon in the deep ocean that lower atmospheric CO2. And the map that you see on the right, all of that red indicates very ancient aging of the bottom waters of the ocean that lock in CO2 so that it's sequestered away from the atmosphere. And I'm in the process of continuing this work to look at how changes in the Earth's tilt influence ocean circulation. I'm now going to pivot to the next project, which is on 1,000-year timescale. So in this project, I'm looking at the last 1,000 years of Earth history, and I'm asking the question, what are the relative importances of natural forcings for ocean carbon uptake on regional timescales? How has anthropogenic forcing changed a system? And what are the implications of this for future climate? And just to orient you, this is an image that shows deviation in temperature from average or temperature anomalies for the last 2,000 years. And the duration of my study goes from 850 AD to 2010. This is the length of the model simulation. And I just want to point out that from 800 to about 1850, this is the pre-industrial period, and temperatures were relatively stable in comparison to what happened after 1850, where we see temperatures exponentially increasing as a result of burning trees and fossil fuels. So I just want to go back here for a moment. These are the natural forcings that drive the system. So prior to human interference, solar forcing, volcanic forcing, land use forces, and greenhouse gas forcing all influence the climate. Now, during the industrial era, what we've seen from this modeling study and from observations is that ocean carbon uptake has exponentially increased. Each of these red lines represents carbon uptake in a region of the ocean. So this focus of the study is regional. It's the second part of a study that looks at global. And this exponential increase, you can see for each of the ocean regions I studied, we see this rapid increase in CO2 in ocean water in response to rising levels of atmospheric CO2. And this is really important because to date, the oceans have taken up 40% of the carbon dioxide that we've put into the atmosphere. And here, this gives us a sense of which regions are important. And the two most important regions for this study are the Southern Ocean and the tropics. These are the regions that are taking up the most carbon. I want to finish my conclusion, sorry. Just a little side conclusion that I don't have time to go into is that another really interesting finding from the study is that the ocean is no longer responding to volcanic eruptions. They used to cause a brief increase in the amount of CO2 that the ocean could take up, but that no longer occurs because of the anthropogenic influence on the oceans. And finally, the third point is the most important, that the the regions that are key for the uptake of carbon are slowing down. And what this means is that the ocean's buffering capacity of all of the CO2 that we're putting into the atmosphere is diminishing. And this is really important moving forward in time. My third project, and Gina, please cut me off if I go over time, is quantifying the impact of invasive plant species on carbon storage in Northeast temperate forests. So this is a modern study. It's looking at how the storage of carbon in trees changes on decadal timescales. And this is actually a picture of my study site. And here you can see a dendrometer. It's a band that measures growth in real time. And forests are incredibly important for the carbon cycle. Globally, they store 7.6 billion metric tons of carbon annually. And in this study, I'm trying to quantify the impact of invasive plant species that come into forests and disrupt ecological processes. Are they impacting the storage of carbon in these trees? And hopefully this type of work will lead to both improved stewardship of forests and also improved models of future carbon storage as we continue to change the climate and change habitats. This is an image of tree cores. So this, in this project, I'm comparing an invaded plot and a non-invaded plot. I'm taking real-time measurements of tree growth, and I also am going back in time by extracting tree cores and looking at their rates of growth. And in addition, I'm sort of complementing this with carbon isotope analysis to look at how water use efficiency varies from plot to plot and also over time. So all of these projects together are a very small piece of a multidisciplinary, international, vast research effort to try to understand, mitigate, and adapt to climate change. And I think that this is one of the most complex problems facing humanity and one that we really need to address soon. And I just have this diagram of all the different, well, just a handful of the different fields that we need to kind of approach and solve this problem. And to be honest, I didn't see that little note at the bottom of the email until today about bringing work into the classroom. But in my 100 level classes, where weirdly I mostly teach psychology majors, I try to kind of bring this to their attention, that they too can be part of the solution. You don't need to be an environmental scientist or a climate scientist. You can be part of the solution from many different disciplines. And also, just generally, I feel like students are most excited when they are exposed to the process of research and how we find the answers and how we formulate questions rather than just being spoon-fed the information that's already in the textbook. So thank you so much for your attention. And I think I'm popcorning to somebody, but I'm not sure who.
Yeah, Amber's next. Amazing. Thank you. And thank you for pointing out interdisciplinary conclusions. That's so important. Beautiful. Thank you so much.
Candace, you're muted.
I just saw that too. I will share my screen, so now I'll work on that. Hmm. Hmm. How everything is moving around, and I haven't been teaching this year because I've been on sabbatical. So now I cannot remember how to share a part of my screen. Wait, let me work on it. Can I share only a part of the screen?
Yeah.
Where
You
is
should be
that?
able to share a window.
If you go to share and then click advanced, then click portion of screen.
Oh, yeah, yeah, yeah. Oh, I see it. I see it. Thank you. Oh my goodness. All right. Man. All right. I think I have it.
It looks
I so like that many of us are talking about and thinking about similar kinds of things, even though we're not in the same discipline. But so many of us are talking about research and that how different kinds of systems interact and how they have an impact on our lives today. So I'll talk about my research in the same way. Some of the new things that I'm thinking about are looking at larger systems. So I will begin by talking about that today. To give you a little bit of background, I study language acquisition in deaf children. And it sounds like maybe a very small field, but it includes aspects from quite a broad, I think, a broad area and is a larger field in itself. Research or people that have been studying language acquisition, that has been happening for a long time. And we've all begun to think that it's something that children do with what they're given. Most of the research is researchers trying to learn how children do that. How do individual children figure out what the language system is that they're in, how it works, because it is very unique and it's quite interesting and it's very complicated to us, this human capability. Most of the research focuses on what happens in the child. And that is also true for a lot of my work as well. So we tend to think about something that language is given to children and then children do something with it. And we want to understand the processes that happen with these children, what they do with what they're given. But that experience of being given a language is not as universal as many people might think. Many deaf children, not all, but many deaf children are born to a family that is not giving them any language. They can't access the spoken language of the parents, and parents haven't learned to sign, so they're not sharing the language. And from those types of experiences that many deaf children go through, it is a unique language experience that can tell us something about how the language of system works for all children, not just for deaf children. So because deaf children have this unique kind of experience, it can give us some information, some ways to understand the process itself that we don't get from studying hearing infants with hearing families. All infants are born with some internal language system. That internal system is not a closed system. It doesn't mean that we understand something fully by just looking at what the child does. We need to understand there are other systems at play, and these other systems are external to the child. So for example, the child's family is external, the community, the culture, even global systems. More recently, I've started to think about some of these broader systems and how we can learn about, or how those broader systems, how they're structured, how they will impact language acquisition for a child. And what does that tell us about language acquisition in general? Deaf children are born with all the same kinds of internal structures that all children are born with. They just can't access these external systems. They can't access the language in those external systems that are being provided. So deaf children, their language experience is unique because of the ways that these larger external systems work and operate and function. These external systems can either operate to support the deaf children's language acquisition, or they can operate to undermine or damage or prohibit their language acquisition. These kinds of experiences are something, again, that are unique to children that are born deaf, to parents that are hearing and do not sign. So one area of my research that I've been working on, and I've been doing this for several years, many years, I ask questions about how children who don't have access to their parents' spoken language, how do they come up with systems of communication? And I've done research, my postdoc research, and my mentor. We had been to Nicaragua to study there to show how language, how do children can create a language from scratch. And there are some important system supports that must be in place there for that language to then emerge. One question that I asked is, what is the form that children's language look like when they don't have access to their parents' spoken language? How do children invent ways to communicate with their family and the people around them? And so I will ask you all to participate now. So if you see these two things on the screen, the one on the left, let's talk about that first. Suppose you had to tell someone to take that thing, pick up that thing, but that person doesn't know your language. Maybe that person is deaf. Maybe you can't use anything spoken. So how might you come up with a way to ask someone for that object? So if you would just do that for me now. Yeah, yeah, that's good. Yes, yes. I like what you're doing. Yeah, those are different things that you could do. This is one. You could do this. Those are all really good strategies. And those are very common strategies. And now, what about the picture on the right? What might you do to get that thing? Right, right. So what you all have showed me right now is exactly what I'm studying with deaf children. Many have used what we term handling. That's the form that you did. You were representing the object by how it is used. So like this. So that means you're handling or holding the object. Or when someone did that, you are handling the utensil, the object. And that is one way to show that. Another way to represent that, and I saw a few of you doing this, is you could use something that represents what the object looks like. So for example, someone did this. So either of these strategies both of them are ways that deaf children would do. And that other strategy is instrumental. So these are the kinds of things that people do when they don't share a language and they want to communicate. So the two ways are handling and instrument, hand shapes. So my research is looking at deaf home signers. They don't have a language, how they decide to choose what strategy of hand shape that they're going to use for their communication. The picture I'm showing you right now is this child. She's 3 years old and a home signer. So that child has not learned Nicaraguan Sign Language yet. She just started attending the school, so she hasn't had access to language. So I sat down with her and was asking her to show me what she does for various objects. So I'm showing you the picture of the saw that's next to her. And so you can see she was using the handling hand shape for that. The finger in her mouth is just because she's 3 years old. That's not part of signing or anything. So what we see in deaf children is they will use a lot of handling strategies, and they will use some instrument hand shape strategies. It seems more common to use handling. Sign languages are different around the world, and they will use different percentages or ratios of instrument versus handling hand shapes. Some use more instruments, some use hand shapes. Each sign language has their own pattern in terms of the use of these hand shapes. Lastly, I think that more recently, I'm interested in looking at the broader issues at play: social context, global context, and how these impact and shape language systems, socio-systems that children are growing up in. These kinds of systems include family systems, community systems, the larger systems such as the economy, the global systems. I've been really wanting to think about these larger impacts on the individual child's language acquisition. So I'm starting to think more broadly about these bigger issues. Deaf children and their language ecosystem really depends so heavily on what happens as part of the broader systems, because they don't have access to language at home. Their language opportunity depends on what's happening in the context of their community, their society, their global system. And I can give you two brief examples. Sometimes these systems are quite supportive for deaf children's language acquisition. Sometimes these systems can really undermine language acquisition. So for example, one example in terms of support is in Nicaragua in the 70s during the revolution. They made new social structures, and that social change, out of that social change, they set up deaf schools for the first time. And these schools for deaf children became places of immersion for deaf children to absorb language and get a language. So the broader community, the politics, the global context of that country then had a direct impact on these children's language experience and development language. And we've been studying that language that they invented at that time. We are still studying that language and how it develops and grows. Another way that the broader systems can really have large negative impact or undermine children's language acquisition is an example that I have last week. I was watching a webinar, and it was a deaf journalist that is in Gaza. And that journalist was sharing their video of what's been happening in Gaza, and he has met several deaf children, and it's on the street, like he notices someone who is deaf and interacts with them. And he has been realized some of the deaf children that were in schools are no longer. So they were in a system that was giving them some opportunity, some support to have language acquisition. But with what's been happening there, those opportunities have been completely destroyed because of the situation in Gaza and what they're going through. A complete destruction of the education system, the community system, the families. So he was meeting children and was recognizing that they are losing some of the language acquiring that they had had in the past. So I am sure that the impact, it depends on many different things, right? The language acquisition depends on the age of the child, how much experiences the child has had in a school for deaf before the destruction started. There are many reasons at play in Gaza, but I do think these issues are important to think about when we are wanting to create or perpetuate things that will have downstream impacts on language acquisition for deaf children. And these are observable impacts that are presently happening, and the deaf journalists reported on in Gaza. And so noting that deaf children are struggling, and that's becoming a more recent area of interest for me, trying to trace these larger global systems and how they impact them and on an individual child's developmental process. So thank you.
Thank you so much. Carmen, I hope you have time to present if you have 10 more minutes.
Hi. Yeah, I already told my students I was probably going to be late. I had my lab meeting after this, so don't worry.
Thank you so much. Thank you so much. And thanks in advance to all the speakers. This has been so amazing.
Sorry, I think I'm doing the wrong share here. Let me just do that. Okay, so PowerPoint. All right, so okay, so can you see that beautiful cell there?
I see the beautiful cell.
Okay, great. So let me just put this back to the presentation. Well, sorry, too many screens here. I don't even know where I am anymore. It's just too much stuff going on. Okay, so presentation. And I have an old computer that takes forever. All right, I think that we are here. So I already introduced myself, and what you are looking at right now is a micrograph, a confocal image of the cell that I study. These are a very specific type of glial cells that are part of our brain and our spinal cord. I think that probably here everybody has heard about a neuron, but these are different type of cells that are also part of our brain and spinal cord. They're actually more abundant as a group than neurons. And they do a lot of things in our brain, but this one that you're looking at that is called oligodendrocytes has a very specific job. And that job is to make what we know as the white matter. And what you're looking at here is a very simplified cartoon of a brain, and this is what the cut of the brain through the middle of it, and we're looking, through the brain cut in this direction. And what you see is that there are two very clear areas that we call the gray matter and the white matter. So the cells that I just show you now, that oligodendrocyte, is what makes the white matter. What the white matter is a very highly specialized membrane that comes from this cell, the oligodendrocyte, and it starts wrapping many, many times in a spiral way around the neurons. So what you see here is actually in this sort of orangey color is the axon process of a neuron. And what this membrane does is basically creates an insulation around the neurons that kind of acts like a plastic around the cables, you know, and nerve impulses are essentially like electricity that is transmitted to the neurons. And that's how our brain, you know, goes to different parts of the body, to different organs of the body, and sends, you know, information for many things. Not all axons had myelin, but for the ones that had myelin, it's very important for the fastest and proper communication between different systems in the body and in our brain. And there are diseases that attack this cell, okay? The origins could be very varied. You know, the ones that we focus in the lab is called multiple sclerosis, which is an autoimmune attack of these cells in the brain. And what you see in this cartoon, in this movie, is that when you lose myelin, it's akin to having a short circuit. All right, so the signals are being generated, but now you have cables that are not transmitting that signal in a productive way. When people start getting these diseases, what happens is that the function, and what you see here is actually coming from human patients, you know, over years, that tell us this sort of like, you know, like peaks and valleys type of outlook, in which some patients will start getting symptoms and the symptoms may kind of relapse, so they sort of go back to being normal. But over time, what's happening is that these attacks where you're having clinical manifestations of something going wrong with the function of your nervous systems, like, you know, optic neuritis. I know that MD, I'm a biologist, but essentially these patients will come to see a neurologist because they know something is off. What is happening essentially is that they're losing the myelin, the transmission of nerve impulses is being problematic now. Because we don't know what causes this disease. We are very limited in ways how we can treat this disease. So right now, essentially these patients are put in drugs that prevent the immune system from attacking, so they're essentially immunosuppressed for most of their life. This is a disease that attacks people in their mid-30s, early 40s, so it's really devastating to get this kind of diagnosis because this is a chronic condition that is probably going to accompany it to the end of your life. And what you see here is that over time, you know, when myelin is lost, the neurons now could also get damaged. So it's not a neurodegenerative disease to begin with, but if you don't have myelin repair, that we call remyelination, eventually the cumulative damage that that disease causes is going to lead to the loss of neurons. And at that point, it's really hard to do something for these patients to recover function. So what we do in our lab is investigating this process of myelin repair or remyelination. And what you're looking at now is a section of a human brain. This actually comes from tissue banks that we get brains from humans. What you see here, and you have to take my word for that because we are not neuropathologists, but that was actually evaluated by a college of mine that are neuropathologists, to help us find those areas where myelin is being lost. So this is white matter, and what you see here, there is like a big gray blob in the white matter, and these are essentially MS lesions. So when we take those parts of the brain and we stain them with a specific, you know, reagents, chemical reagents, what you see here, these nice dark blue circles, is indicative of good myelin, healthy myelin. And when you see that demyelination has happened and you look at the same and these areas, what you see are the axons of the neurons that are still there. This is an early lesion, and you can appreciate that the myelin has gone away, so we don't have myelin there. But early on the disease, and this is why the reason why you see relaxes, the cells in the brain, the oligodendrocyte precursor cells, could actually come and try to put myelin again around those axons to restore function. And this is the process of repair called remyelination. And what you see here is essentially this outcome, okay, in these schematics, in which oligodendrocytes will essentially, new oligodendrocytes will come and try to repair those pieces of myelin that has been lost from those neurons. The problem is that that process gets worse and worse as the disease progresses. And as I already mentioned, if we cannot get myelin back into those axons that were myelinated to begin with, what you get is neurodegeneration. So what my lab does is trying to understand what are the signals that can actually help those oligodendrocytes that are in the brain and could actually remyelinate, do this job better. And in the process, we actually need to deal with what are the factors in the cell, what we call cell autonomous, right? Things that are happening in the cells that may be preventing to do their job versus versus things that can be happening in the environment that we call non-cell autonomous that may also impact the ability of those cells to do their job, okay? And this is multifactorial. This, of course, are like all things in life and complicated, right? But we have been investigating for the past 10, 15 years in the lab how the mechanical properties of the brain and the spinal cord change in the context of demyelinated brain injury. This is a very fancy word. It's called mechanotransduction, but essentially what it means is how the cells interpret the mechanical signals of the environment and convert that into some kind of biological response that is important for repair or for development, okay? Mechanotransduction happens normally during development, and we argue that this is something that also has to be important for the process of remyelination and repair. And there are many things in terms of mechanics that can happen in tissues, and this is just like an schematic to show you this kind of forces. Not all cells in the body are going to be presented with all of these forces in a particular time of their life. But what we've been sort of focus is on this property of elasticity or stiffness, which is theoretically how soft or stiff that area around the lesions feels, right? We discovered a few years ago, this is experimental approach that we use in the lab for telling how we do it. Right? I'm going to tell you how we do it and tell you what the data is. So as you show, we use actual brains, and those brains can come from humans or from animal models, from mice, okay? We are able to purify cells from these organisms, in the case of mice and rats. We also have ways to get cells from humans that I'm going to mention in a minute. And we could actually put those cells in culture and make co-cultures of axons and glial cells together to actually look at control environments where we can change particular properties and see how that impacts myelin formation, which is what you see here. This is essentially green lines and the blue lines. The blue lines are neurons, are axon and neurons. And then the green is actually the myelin around those axons in vitro, in actually, cell culture that we can do here in the lab. And what you have here at the bottom is actually hydrogel, which is a way to mimic the environment of the brain. We can actually make those hydrogels in ways that can make feel like a normal brain versus in ways that can feel like a lesion brain, okay? So what we did a few years ago was actually take brains from mice and do things to those brains with chemicals that make them lose the myelin. And we actually look at an area of the brain that is called the corpus callosum that is heavily myelinated, and essentially communication of the two hemispheres of the brain. And what you see here is what that looks like after we do these kind of treatments. So this is myelin in green, and after we give those particular treatments, the myelin goes away. And what we did is that we took a microscope called an atomic force microscope that essentially allows you to push, okay, using a laser, to push into these areas of the tissue. And the amount of force that you put in that thing is going to be returned to you and tell you, how soft or stiff that area is. So we did that, and we discovered that when you demyelinate the brain in an acute manner, okay, the stiffness of the brain goes down. And this is essentially the color code. The warmer the colors, the stiffer the brain, the cooler the colors, the softer the brain. So in a normal brain, you are around here. And then when you do acute demyelination, when you lose myelin, the brain becomes softer. There are ways to manipulate those systems in which you allow time for recovery. And that's remyelination. And then when we do that, let me just go into the next slide, okay? What we discover is that if we allow this system to recover, the stiffness of the brain kind of comes back, not to exactly the same point that it was before demyelination was induced, but it recovers some of the stiffness of the control conditions. But then if we do another different protocol in which we can simulate the accumulated damage of chronic demyelination, what we found is that the brain becomes actually quite stiff, all right? And we argue, based on some of the other data that we had before, that stiffness of the brain is one of the factors that it may be contributed to the lack of remyelination in this kind of chronic injury in the brain. And we actually did that in mice, but we were actually able to look at human samples as well. And what you're seeing here is actually coming from brains of patients that die of multiple sclerosis. And what you see here is essentially that this is about the level of what we call normal appearing white matters, areas where the brain appears to be normal amount of myelin. And these are essentially many samples with using this atomic force microscopy that tell us that within the brain of humans, we can find lesions that are stiffer than the normal appearing white matter, or they are softer than the normal appearing white matter. I have a lot of data that I cannot present to you right now because we don't have the time, but the conclusion is that within the lesions of the brain, we can find, indications that these softer lesions are compatible, histologically speaking, with early acute lesions of demyelination that we know can actually hopefully improve and remyelinate, whereas these guys here that are stiffer are actually compatible with chronic demyelination, okay? So we found, as I said, these both in models of rodent models and humans, and then we wanted to see, because we already have some data with the mice, if we were to be able to take cells from the brain, human cells, and test it into an in vitro system, whether we actually will see any effect in their ability to make myelin in this kind of simplified systems. Because
Carmen, you have 1 minute to wrap.
Yeah, I'm about to finish. So what we did is we actually got from the Miyares Foundation cells, that had been reprogrammed. This is what is called induced pluripotent human stem cells technology. So you can take any cell, normally they're taken from the skin or the blood, and you can reprogram them into becoming any type of cells that you want. So we did that. We got several human cell lines from the Miyares Foundation. We reprogrammed them into becoming oligodendrocytes, and we measure what will happen to them in terms of differentiation and myelin formation when we expose them to these different conditions of softer versus stiffer brain. And this is what we found, essentially, that when you look at these cells in the context of healthy brain, we get a lot of cells that are oligodendrocytes, and those cells actually differentiate. This is essentially markers to tell us that these cells are differentiated, and these are actually markers that tells us that these cells are oligodendrocytes. And what you can appreciate here is when we have these cells in chronic conditions, those two factors go down. They become less, less of that. And, this is essentially statistically significant for the chronic demyelinating conditions, whereas for the acute, we have a little bit of, mix back there that we're still trying to figure out why these differences are. But these are 3 different patients that we got, 3 different donors that we got, and we see the same trends. So essentially, that's what we're trying to understand. What are these difference in the cell behavior that are prompted by the difference in the mechanical properties of the brain, because we believe that this is very important to promote repair of these lesions, because it doesn't matter that you have very good cells and those cells go into an environment that is not productive to make them be able to do their job. So that's it.
So much. Thank you so much, Carmen. Thank you so much, everyone. I just want to point out what incredible work women in STEM are doing at Hunter. Thank you for the LUCE Foundation. Your research means so much to all of us. So thank you for your incredible work. Thank you for presenting it at ACERT. We hope you have a beautiful, beautiful rest of the day. But thank you. That was incredible. You are all doing such incredible work. Have a great rest of the day. Thank you. Thank you. What amazing research. Go women in STEM. I love it. Makes me so happy. Take care, everybody.
Thank you. Thank
Bye.
you. Thank you.